Hello students, and welcome to Crop Science 6049. In today's lesson, we'll be looking at cell division whilst focusing on meiosis. By the end of this lesson, you must be able to describe the process of meiosis, explain the significance of meiosis in plant reproduction, and compare and contrast meiosis and mitosis. So let's take a look at meiosis. Meiosis is a nuclear division that involves the reduction in the number of parental chromosomes by half and consequently the production of 4 haploid daughter cells. The process of meiosis takes place in 2 stages, namely meiosis I and meiosis II. During meiosis I, a cell is divided into 2, and in meiosis II, even further division takes place, resulting in a total of 4 haploid cells. It occurs in the formation of ovules and pollen grains in plants. It takes place in anthers and ovaries. Before the start of the process, the parent cell goes through the preparation stage of interphase, where it goes through the G1, S, and G2 stages. During this phase, the parent cell synthesizes more DNA and proteins, thereby increasing the overall size and mass of the cell. Let's take a look at meiosis I. Meiosis I starts with prophase I, and during prophase I, the following events occur. Chromosomes become visible because of shortening and thickening, and each chromosome is seen to consist of a centromere. Chromosome pairing, also called synapsis, between homologous chromosomes occur, and each pair is called a bivalent. Homologous chromosomes appear to repel each other and begin to move apart at the centromere. Homologous chromosomes join at several points along their length called the chiasmata. As the chromosomes move apart, the chromatids sometimes break at the points where the chiasmata occurs, and there is exchange of equivalent sections of chromatids between the homologous chromosomes. The exchange is called crossing over. Crossing over is the interchange of corresponding chromosomal parts between sister chromatids of a homologous pair of chromosomes, resulting in recombination of genes. The chromosomes further contract and thicken towards the end of prophase. The nucleolus disappears and the nuclear envelope also begins to break down, and this marks the end of prophase. Now let's take a look at metaphase I. During metaphase I, the following events occur. Bivalents arrange themselves at the equator of the cell attached by their centromere. The spindle contracts slowly, pulling centromeres apart. Let's take a look at anaphase I. During anaphase I, the following events occur. Spindle fibers contract and pull homologous chromosomes apart. This separates the chromosomes into 2 haploid sets, one set at each end of the spindle. Now let's take a look at telophase I. During telophase I, the following events occur. The homologous chromosome pairs complete their migration to the 2 poles. Now a haploid set of chromosomes is at each pole, with each chromosome still having 2 chromatids. A nuclear envelope reforms around each chromosome set, the spindle disappears, and cytokinesis follows. After cytokinesis, each of the 2 progeny cells has a nucleus with a haploid set of replicated chromosomes. Let's take a look at meiosis II, starting with prophase II. During prophase II, the following events occur. Nucleolus disappear. The nuclear envelope breaks down and the spindle fibers form. Poles are at right angle to the plane of the previous cell division, and the spindle develop at right angle to the spindle of the first meiotic division. Let's take a look at metaphase II. During metaphase II, the following events occur. Single chromosomes align themselves at the equator of the spindle as chromosomes do in mitosis. This is in contrast to metaphase I. in which homologous pairs of chromosomes align at the metaphase plate. Spindle fibers are attached to the centromere of each chromosome. Let's take a look at Anaphase II. During Anaphase II, the following events occur. The centromeres separate and the two chromatids of each chromosome move to opposite poles on the spindle. The separated chromatids are now called chromosomes in their own right. Now let's look at Telophase II. Telophase II is identical to telophase of mitosis, with no notable differences except the position of the metaphase plate and production of 4 gametes with distinct genetic information and many possibilities for offspring resulting from sexual reproduction. During Telophase II, the following events occur. Chromosomes uncoil and become invisible. Nucleolus reform. Spindle fibers disappear. Each nucleus possesses half the number of chromosomes of the original parent cell. A nuclear envelope forms around each set of chromosomes. Cytokinesis takes place, producing 4 daughter cells, each with a haploid set of chromosomes. Because of crossing over, some chromosomes are seen to have recombined segments of the original parental chromosomes. Now, what are the importance of meiosis? It is a source of genetic variation because of crossing over, independent assortment, and random fertilization. It is important for genetic variation since meiotic products are non-identical because of crossing over and random assortment of chromosomes. It is important for sexual reproduction, which involves the production of ovules and pollen grains in plants. It is important for breeding purposes, e.g., to produce better varieties of crops. What are the differences between mitosis and meiosis? Mitosis consists of 1 nuclear division, whilst meiosis consists of 2 nuclear divisions. In mitosis, 1 cell cycle results in the production of 2 daughter cells, whilst in meiosis, 1 cell cycle results in the production of 4 daughter cells. In mitosis, the chromosome number of daughter cells is the same as that of the mother cell, that is diploid. In meiosis, daughter cells contain half the chromosome number of the mother cell, that is haploid. In mitosis, daughter cells are identical with mother cell in structure and chromosome composition, whilst in meiosis, daughter cells are different from mother cell in chromosome number and composition. Mitosis occurs in somatic cells, whilst meiosis occurs in reproductive tissues. In mitosis, there is no pairing between homologous chromosomes, whilst in meiosis, homologous chromosomes pair. In mitosis, crossing over and recombination do not occur, whilst in meiosis, crossing over takes place. In mitosis, chromosomes are in the form of dyad at metaphase, whilst in meiosis, chromosomes are in the form of tetrad at metaphase. In mitosis, at metaphase, centromere of each bivalent divides longitudinally, whilst in meiosis, the centromere does not divide at metaphase. In mitosis, 1 member of sister chromatids move to opposite pole during anaphase, whilst in meiosis, 1 member of homologous chromosomes move to opposite poles during anaphase. Mitosis maintains genetic stability due to lack of segregation and recombination, whilst meiosis generates variability due to segregation and recombination. Now let's take a look at the similarities between mitosis and meiosis. Mitosis and meiosis are 2 mechanisms involved in the reproduction of multicellular plants. Both mitosis and meiosis starts from a diploid parent cell. Both mitosis and meiosis are processes of nuclear division of cells. The major steps of both mitosis and meiosis are interphase, prophase, metaphase, telophase, and anaphase. The DNA of the parent cell is replicated prior to nuclear divisions. Mitosis is similar to meiosis II. During the metaphase of mitosis and metaphase II of meiosis, individual chromosomes are arranged in the cell equator. During anaphase of mitosis and anaphase II of meiosis, sister chromatids are separated to opposite poles. Both mitosis and meiosis end in cytokinesis. Thank you for watching. Don't forget to subscribe to this channel. If you have any questions, additions and subtractions, please write them in the comment section. If you feel you have benefited from this video, click the like button.